So we're reviewing, good, you guys are already in it. Phew. So my first question was, how many of you have had organic chemistry already? You may have had it in the past, you may not have had it at all. You may be taking it right now. A lot of knots, okay, so this should be new for many of us, okay. So we're gonna do some review and I'm gonna do some questioning during this. So this is just to see what you picked up from Monday. What name is given to a molecule that contains carbon atoms bonded to other atoms? So, or other carbon atoms. So this one you'll write in the answer. So carbon bonded to other carbon creates organic molecules. It used to be we would say all organic molecules, well, it is true, all organic molecules contain carbon, but not all carbon molecules are organic. Like carbon dioxide or carbon monoxide, they are not organic molecules. So the key is that it has to be bonded to other carbon atoms. So why is a carbon atom ideal for building molecules of life? So most of you have had chemistry in the past and you know about the valence electrons. The valence electrons are the outer shell, right? They're the available electrons for reactions. We know we want to fill the outer shell. The outer shell can have 8 and they have 4, which means they're very versatile with what atoms they can bond to. They can form single bonds, double bonds, triple bonds, meaning they share 1, 2, 3 electrons with other atoms. They can form long chains, which can be used for stability, rings, branches. So that structure of 4 makes them very versatile. So they create very strong, stable bonds, like the single bond, the double bond, the triple bond. The more bonds you have, the stronger that bond is, and they are all covalent bonds. I saw somebody had put that. So there's no limit to their size, and I kind of mentioned the other 2 already. So here's another question for you. Which is not an organic molecule? So it gives you lipids, carbohydrates, carbon dioxide, proteins, and I think it gives you nucleic acids. So I guess I have to hit start for that. So you guys should be, I'm sure, real quick at answering this one. So the carbohydrate, or the, carbon dioxide was not the organic molecule. That was actually an example I gave, so I should have maybe given you something else, because it's not bound to other carbon atoms or hydrogen atoms. So all organic molecules kind of start out as a hydrocarbon chain, and then you're going to remove hydrogen atoms and put in functional groups, and that's what you were looking at on Monday. So the 4 organic molecules we want to be really familiar with throughout the whole year are the carbohydrates, the nucleic acids, the lipids, and the proteins. And you've heard a little bit about them already in 9th grade. So what name is given to an organic molecule that consists only of carbon and hydrogen? So this one you're going to type in. So the name gives it away. Hydrocarbon is made of carbon and hydrogen. I saw carbohydrate as one of our choices. That is going to include oxygen as well. So it has those hydroxyl functional groups. Don't worry, these aren't going to be nonstop question after question. I think a content slide will come next. What name is given to small specific groups of atoms that can attain be attached to that hydrocarbon chain, the carbon skeleton? Okay, so functional groups, which is the topic that we're talking about. So we have our hydrocarbon chain, right? That is going to change its functionality once you put a functional group on it. So I usually have a quiz over memorized functional groups. I don't know if I should do a quiz over this one or not. We could do a quiz next week if you wanted in a lockdown browser. Just these short memorized things that I know it's kind of had over the internet. But functional groups give molecules their unique shape. And remember, the whole year, we're going to concentrate on that theme that the structure determines the function. So the functional groups give it its shape and therefore give it its functionality. And when we say functionality, I'm referring to like what kind of chemical reactions they can undergo, or how they can be used in the living organisms. So here we got another one. We got functional groups. I'm giving you 2 minutes on this one. So I've put in structures, and I want to see if you can match them with their proper name. So this is what you want to identify. Hydroxyls, the OH has a negative charge on it. So OH, hydroxyl, you'll see those in carbohydrates. You'll see them also attached to some of your hydrocarbon chains in amino acids. The carbonyl, I didn't give you a straight up carbonyl. The ketones and the aldehydes are both carbonyls. You guys are seeing the screen while I'm seeing it, yes? Okay, thank you. So the difference is the position of that double bond O. So in aldehyde, the double bond O will be on either the first or the last carbon. So it's like a bookmark. And the ketone, the double bond will be somewhere in the middle. So I think of the alphabet, and the A is at the very beginning of the alphabet. And that helps me remember that the aldehyde is at the very end of the chain. Ketone, K, is somewhere in the middle of the alphabet. So that helps me remember that the double bond is somewhere in the middle of the chain. Phosphates, that should be one that we are familiar with from nucleic acids and phospholipids. We talked about those. It's also the only one with a phosphorus, so that should be easy. The amine group, N, sometimes you'll see it with 2 hydrogens, sometimes you'll see it with 3 hydrogens. It depends on what it has attached to it. So you might not have seen the R before. That's the basic hydrocarbon chain, and it's miscellaneous. So it could be like a functional group. It could be the main skeleton. So that's like a variable, but consider it to be any of your carbon atoms, basically. The amine, the methyl group, CH3. Sometimes people confuse the methyl group with the end carbon atoms, because if they don't have a functional group, they're going to have hydrogen attached to them, but they're still part of the basic chain or the basic ring. They're considered a functional group when they're hanging off of that ring or they're branching off of that chain. Sulfhydryl, you'll find those in amino acids, really only one amino acid, cysteine, and so it's the only one with the S, the only one with a sulfur, so that should be easy. So let's keep going. This hydroxyl group, this is the OH that I'm talking about. You can see it hanging off of these structures. So we're going to talk about our 4 macromolecules, sugars, where we're going to see this. It makes things very polar because the oxygen, you know, is electronegative. So it's attracting the electrons toward the oxygen molecules, which creates a polarity in molecules. And we know polar things like water, so it makes them hydrophilic. If you haven't seen a structure that did not contain carbons, in organic chemistry, this is kind of shorthand writing, where 2 carbons come, like you'll use just lines, and where 2 lines come together, there's a carbon atom. So they don't, because organic chemistry is all about carbon, they don't bother writing in all of the carbon atoms. So when you see like some stick figures, just assume wherever 2 sticks come together, there's a carbon atom there. Okay, so the hydroxyl group. And then the carbonyl group is the double bond O. Here's showing you 2 different molecules with carbonyl groups. This one has its carbonyl group on the end, and this one has its carbonyl group somewhere in the middle. These are both sugar molecules. So the position of that carbonyl group makes for different functionality in the sugar molecule. So here we can see it's in the middle, so it would be a ketone. And here we see it's on the end, so it's an aldehyde. And again, that double bond O, highly electronegative, so it makes the molecule very polar and therefore hydrophilic. Carboxyl, our C double bond O, OH, it makes like carbon dioxide dissolved in water, you know, is carboxylic acid. So this has an acidic property in that it can donate hydrogens in solution. So that makes it acidic. So when we're looking at proteins and lipids, we refer to amino acid and we refer to fatty acids, and that acidity is coming from that carboxyl group. So amines are here, your NH2, they are ionic because they can gain or lose hydrogen atoms and therefore gain or lose their protons. And so that creates the ability to be an ion. So when we get into our proteins, we have 20 different amino acids. They all contain amine groups, but different hydrocarbon chains and different functional groups are attached to them, giving each of those amino groups or each of those amino acids a different property. Sulfhydryl, the SH, so again, R is just a variable amount of carbons, so it doesn't matter, the functionality is here. So this forms a disulfide bridge. When we talk about protein structures, we have 4 different levels of structures. Our amino acids, our primary structure, the local folding is secondary structure. Tertiary folding is like whole group, the whole sequence is folding in together, and sulfhydryl bridges play a big role in stabilizing that tertiary structure. So here we see just the sulfhydryl group, and here we see that disulfide bridge. So we're going to talk about proteins next week, so we'll get into the sulfide bridges a little bit more. The phosphates, you're going to see them in your nucleic acids. ATP, we learned about nucleotides in 9th grade, right? Guanine, adenine, cytosine, and thymine. They each actually have a triphosphate form. You're just only currently familiar with the ATP because we talk about it a lot with energy. But they each have phosphates here. Remember, they're on the outer structure of the DNA molecule or on the outer structure of the nucleotide. We also have them attached to our fatty acids. And remember our phospholipid bilayer that surrounds most of our organelles as well as our cell membrane as a whole. So remember, you had hydrophobic fatty acid tails and hydrophilic phosphates. So these are your hydrophilic portions. Okay, so when we look at molecules, which is what you're going to do today, you're going to look at those functional groups. If you see an amine group, you know you're looking at an amino acid or a protein. If you're looking at something with a whole bunch of hydroxyl groups, most likely you're looking at a sugar. If you see something with phosphorus in it, you know you're looking at a nucleic acid. And if you're looking at something with a sulfur, you know it's a protein. So when looking at these molecules, we want to look at the elements that are in them. Notice they all have CH and O, carbon, hydrogen, and oxygen. So we're not going to be able to limit it from that. But if they only have carbon, hydrogen, and oxygen, we know it's either a lipid or a carbohydrate. So carbohydrate means carbon with water attached. So this has a 2-to-1 ratio. You know, water is H2O. So carbohydrates will always have a 2-to-1 ratio with hydrogen and oxygen. Lipids do not have that ratio. Lipids will have way more hydrogens than oxygens. So if you see a molecule and it only has CHO, we know it's one of these two. Then I'm going to count up my hydrogens and compare them to the oxygens. 2-to-1, it's a carbohydrate. Greater than 2-to-1, it's a lipid. If it contains a nitrogen, it's either proteins or nucleic acids. So now look at the functional groups that are attached to that. If it has a phosphorus, we're going to name it a nucleic acid. It may or may not have sulfur, in which it's a protein. It will have that amine group that we looked at, the NH2, NH3. So when I'm looking at these, I see nitrogen here, no nitrogen here or here. So I'm going to count up my hydrogens on these two. I know these two are my carbohydrates and lipids. 1, 2, 3, 4, 5, 6, 7, 8 hydrogens, 3 oxygens. That is not 2 to 1. So this would be a lipid. This is your glycerol. Over here, 1, 2, 3, 4, 5, 6 oxygens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 6 to 12 is a 2 to 1 ratio. So this is your carbohydrate. These two had nitrogen, so they're my amino acids or my nucleic acids. This one has a phosphorus group. Here's my phosphates. So I know that this is a nucleic acid, nucleotide. It's a guanine triphosphate. And then this has my amine group right here, as well as a carboxylic acid. Both of those things are present in amino acids. So I know this is an amino acid and therefore part of a protein. It's not an entire protein. Okay. So what questions do you have right now on any of this or on Monday's PowerPoint? Go ahead and unmute yourself if you have a question. It's a lot easier for me to hear you than, I know most people like the chats. I like your voices. I'm finding the chat. There it is. See, I ignore the chat a lot because I'm always hiding it out of my way. Does carbon dissociate in water or not? It does. The carbon itself doesn't dissociate. In organic molecules, they won't dissociate in water. They'll stay as one, which is going to be important in calculating water potential. Okay, but hopefully you got the information that you need in order to complete this worksheet. So the worksheet is linked in your classroom. You're identifying organic molecules. So you're basically identifying the functional groups, and then based on the functional group, what type of molecule would it be? So that was like this, when we did this, we see all these hydroxyl groups and this carbonyl group. So I know that this is a carbohydrate. So I would circle my carbonyl group and my hydroxyl groups. Okay. Okay, you should be watching the daily videos each day. Today's daily video is on properties of biological molecules. So, you know, as we start talking in more detail, what makes living organisms is molecules, and so the functionality of those molecules will affect most of the processes we talk about from here on out. So I will go ahead and let you click off if you're done, or shout at your friends, say hi. Nice to see you, missed you, all that jazz. And I'll stay on in case you need some help with anything.